Start the session now. Yeah. I hope the screen is visible for all of them. Ma'am, could you please reshare the PPT, ma'am?
It's not visible?
No, ma'am, just now the recording started and that's why. Would you please reshare?
Okay.
Yeah.
Can you see now?
Yes, ma'am, it's visible. Thank you, ma'am.
Yeah.
So, well, I would like to give a small introduction about our speaker today. Dr. Hemavathi has completed her PhD in molecular biotechnology from Konkuk University, South Korea, MSc in microbiology from University of Mysore, and BSc from University of Mysore again. She has been awarded as a Junior Research Fellow, Postdoctoral Fellow, Senior Research Fellow, Principal Investigator, and also as DST Women Scientist from UAS and GKVK. Also, she has got Postdoctoral Fellow from National Academy of Agriculture Science, South Korea. Dr. Hemavathi has made significant contributions in plant sciences by publishing more than 15 research papers and attending conferences and presenting posters. She is now currently working as an assistant professor in the Department of Biotechnology at REVA University, Bangalore. Today we have her talking about molecular farming, mainly stressing on using plants as a bioreactors. Welcome to the session, ma'am, and over to you, ma'am. Thank you.
Thank you, Dhanashri. Good morning, one and all. Can you all see the slides? Is it visible?
Yes, ma'am, it's visible.
Okay. Oh. Thank you very much.
Ma'am, could you please put it on the slide share mode?
Yeah, I have transferred.
Okay, okay, ma'am.
Yeah. So today's webinar, I am going to present about a consolidated version of molecular farming, where we will discuss about farming of plants in a different way, where we can use these plants as bioreactors. So whatever farming we do for growing seeds or leafy vegetables for food grain purpose, for edible purpose. But however, here we can also harness the effectiveness of these plants for the benefit of mankind by expressing various pharmaceutical products. So today we shall have a discussion on this. So what exactly molecular farming is? So these are basically agricultural plants that are used for the production of useful molecules that are non-food, feed or fiber applications. So we used to have, as I told earlier, we use this for growing for grain purpose, but however, we can also use them for growing non-food purposes, feeds or fibers, various fibers applications. So basically the important thing that we are going to use these plants is for growing or making plants to express this genetically modified organisms in plants, which in turn make the plants to accumulate these proteins such as therapeutics and diagnostic proteins, and also industrially important enzymes. So these things are usually developed in industries, but also people have extended in using the.
Excuse me, ma'am.
Yeah.
Ma'am, sorry to interrupt, but the slides are not changing, ma'am.
Not changing, huh?
No,
No,
ma'am.
you're actually sharing the window where it's not yet in the presentation mode.
Okay,
So
is it?
can you please stop sharing and share the presentation window or maybe the whole screen?
One second. So is it visible now? Hello? Is it visible?
Ma'am, no, ma'am.
No, ma'am.
Is it changing?
Did you stop and reshare? No?
Yeah.
Okay.
Is it
Yes.
visible now?
It's visible. Can you go to the next slide?
Yeah. I have changed the slide.
Okay, we are seeing the slide number 2. What is molecular farming in plants? That one.
Yeah. Now it's slide 3.
No, I
No?
think either there is a delay or yeah, correct. There is a delay. That's all. Okay. You can
Okay.
continue sharing this, but there'll be a small few seconds of delay. Not an issue. You can continue sharing.
Shall I continue?
Yeah, please continue.
Yeah. So therapeutics and diagnostics diagnostic proteins and industrial enzymes can be produced using these plants. So basically, these are the plants which are genetically engineered so that we can change, modify according to our needs. So let us discuss. So what are the different approaches scientists use, and what kind of systems where we can grow these plants, and what are the different kinds of products that have been released till date, and then what are the advantages and disadvantages or limitations and the challenges and threats that are posed by molecular farming. So now I have changed the slide. Can you see? Hello?
I'm still in the second slide.
Yeah, still in the second slide. I have gone to move to third slide.
No.
No? Shall I share once again?
Ma'am, I think
Yes,
you can
ma'am.
continue. Maybe if there is a delay, we can see, ma'am. If it doesn't change for the fourth slide, then I think you can reshare.
Okay.
Yeah.
Okay.
The slideshow is not visible. Only the
Now it's visible?
no, that full screen thing is not
Okay.
visible.
Yeah,
Only
yeah.
PowerPoint is visible.
Yeah. Okay. Now same. It's getting paused.
Yeah, I think it's a different screen, but that's why.
Ma'am, can you try sharing your whole screen
Okay.
when you do share?
Yeah, this is my new screen. Now is it visible?
No, ma'am.
No.
I think this is only fine. If you could increase the zoom and
No, this is fine.
this
But if you can,
screen itself is fine. Instead of
This is fine,
slideshow,
huh? This is fine.
instead of slideshow, you can change the slide here itself.
Yeah. See, now it's moving, right?
Yeah.
Okay, yeah. When I go to slide mode, it's stopping.
Correct. Yes.
Yeah. So basically, genetic transformation, all of you are aware, whoever belongs to science category. So they all are aware of recombinant DNA technology. So what we do is here is just select a particular gene of our interest and modify and put it into the expression vector system, then introduce it into the plants through agrobacterium means, and then developing these plants in GMO fields. So these GMO fields cannot be grown wherever we want. They should always be away from our ecosystem, and then we have to grow them, particularly mention, and should not be allowed other native plantations should be grown along with these GMO plants fields. So and then harvest whatever fruits, vegetables, grains, pulses, whatever we are intended to, and then the next step is we have to go for extraction, purification, and then packaging and release it into the market. So this is the overall basic structure of genetically modified or genetic transformation. So now we shall see what are the different kinds of products that can be released, synthesized in the plants, and then they can be released into the market. So basically, as we can see here, there is a tree which can be considered as bioreactors, where we can target the production of these pharmaceutical or commercially valuable pharmaceutical products as a whole system. We can express it in the whole plant or a part of the plant, or for a short period of time or for a very long period of time. We can use different strategies. So basically therapeutic products are there, nutritional components are there, vaccines are there, biodegradable plastics are there, industrially important products such as enzymes and other diagnostic kits. So all these diagnostic reagents in the form of enzymes can also be produced. So, and also oils, waxes can also be produced. So that is the reason why these plants are known as bioreactors. So here specifically, what are the different kinds of plants that have been used till now for production of these therapeutic products, such as for immunonutraceuticals, we can use safflowers, rice, and then barley and Arabidopsis for the utilization or production of therapeutic human proteins. So carrots have been used, duckweeds, which is an aquatic plant, and this safflower and rice. So tobacco. So they have targeted both monocots as well as dicots. They have targeted both edible and as well as non-edible leafy vegetables. So and then vaccines, when it comes to vaccine, tobaccos, potatoes are mainly targeted because these tobaccos, why they use this, because they are very good biomass producing agents. Okay, so they can very short period of time they can grow robustly. So the more the biomass, the more the therapeutic product production. That is the main reason why they use tobacco. And potatoes are also a rich reservoirs, so they are very good. I don't know, in India we never use potatoes. We cannot use it in a raw way, but in foreign nations, many people like to eat potatoes in the raw form. So that is also one reason they can directly take potatoes if they are expressing edible vaccines. So which can be used as oral immunization. So coming to antibodies, tobaccos, and then again potatoes, tomatoes, all these things can be used for various purposes. So overall, the main ingredient of this molecular forming is choice of a plant, where in a short period of time, where they can accumulate large number of or huge number of these therapeutic products. And the biomass should be good, adaptability should be good, and their response to pathogens should be good. All this will be taken into consideration. So the strategies are, so they are known as basically green factories, where, as I told earlier, whole plants can be used, and also microalgae. Microalgae, these are evolved eukaryotic microorganisms, where it's very easy to modify the genomic stature of the microalgae. So we can target both nuclear transformation as well as chloroplast transformation. So there are both benefits of nuclear transformation as well as chloroplast transformation. Similarly, aquatic plants. Aquatic plants, they're like just like we just put a fresh piece of duckweed within 5 to 6 days, they grow robustly. So this is one major property that is used for the production of these therapeutic proteins. And then plant cells can also be produced in the form of hairy roots. Hairy roots are really very good. Like we can just grow them very rapidly, and then just in a small bioreactors. And the accumulation of protein is very huge when we use hairy roots. That's why many people are targeting for induction of hairy roots, and it can be induced by any kinds of tissue of any part of the plant. So, and one more thing is so development of suspension cultures. Suspension cultures are also be developed, and then the production rate of these therapeutic products also be high. So when we are targeting whole plants, so it is important that what kind of target we have to look for, whether we have to go for transient expression, means it's for a short period of time, stable expression, it's for very long period of time, but the scale of production will be very faster. So transient expression basically involves plant virus based, expression vector based, and the nuclear stable expression always have to be applied directly to the nuclear or chloroplast. And then whether we need the accumulation of these therapeutic products in leaves or in seeds, that we should look for, because at the end of the day, extraction, once even the proteins are accumulated in the plants, the extraction and high amount of protein production is important. So that is why leafy vegetables are very good in biomass in nature. Seeds, sometimes environmental factors play a major role in the yield, so it contributes much so for the production of intended proteins. So that also we should keep in mind. And then we should go for all, once we decide all these strategies, a particular protein, a particular system, then we should go for designing our gene of interest. So here, this is a consolidated list of comparison between production platforms. There are various production platforms as shown in the slide. One is plant cells, transgenic plants, a plant virus system, animal cells, transgenic animals, bacteria and yeast. Right now, whatever therapeutic products that are produced, majority of these products are produced using mammalian system. Why? Because they are very fast. But however, there are both pros and cons. So that is why what they did is, people, when the first people, US companies started producing these therapeutic products for a disease called Gaucher disease, they used this Chinese hamster cells. So, but however, later they encountered a lot of contamination, okay, from the animal-derived viruses. So, and they lost a lot of money in that also. So, final cost effectiveness is the major role that plays in the, that is in the this therapeutic protein production. That is why whenever you see even a normal insulin which is produced by recombinant DNA technology is very quite expensive when compared to a normal insulin. So that is why people started looking for different sources where they can generate these proteins in a more cost-effective way. So in such cases, animal cell system and transgenic animals were always expensive because their maintenance and cell culture systems and animals, they have to be taken care of very well, because lot of contamination chances will happen, which are derived from both animal as well as human viruses. But in plant cells, when we look into the overall cost, plant cells are medium, and also transgenic plants are medium as well as cheaper. And also the major important in therapeutic or biopharmaceutical productions is good manufacturing practices. So when we are looking for good manufacturing practices, and when we are doing it in animal cells or transgenic animals, bacteria and yeast, the first step itself, it should start good manufacturing practices. Means from the point one, from point A to Z, everything should be followed. Standard protocol should be followed. Everything should be maintained. But however, in plant system, from the designing to expression growing stage, no good manufacturing practices can be followed. Okay, so that is one major usefulness of this using plant system. So here, after growth, so in such cases they can save a lot of money, and it's also very cheap and less low labor cost, and also very and also after they produce these proteins, then they have to start their good manufacturing practices. So like this, when we see the overall cost, scale up capacity, production cost, and also the one more important thing is protein folding accuracy. So once the protein that has been produced in the transgenic plants, is both animals as well as these humans as well as plants or eukaryotic system, protein folding accuracy will be very high, and also the product quality, and then yield, and then storage cost, safety, public perception of risk. So when it is plant derived, people are always like they go for plant based. They don't find any risk in plant based. So if they are produced from animals, they will have certain apprehensions. But whenever it is plant derived, they don't mind taking it and directly having it. So like this, production, vehicles, and then multimeric protein production, storage units, and glycosylation. So all these things, when we take it into account, so transgenic plants are more congenial and also cheaper and cost effective. So in this case, that is the reason people started producing transgenic plants. So the recent update is people are like using hydroponic system instead of growing in the field with lot of constraints, because pros and cons are always available. So plants are going for hydroponics where they can easily grow in a limited place, and also in a very controlled conditions, and also they are very cheaper comparing to, because when you grow in the field, lot of environmental factors will be involved which we cannot control. So that is one of the most important things when we use plants for this as bioreactors for production of therapeutic proteins. So here, coming to different kinds of approaches, how we can express these genes for the production of these therapeutic proteins. One is stable transformation, plant cell suspension culture, and then viral vector mediated transient expression. So as we can see in the picture, this stable transformation can be targeted both for nuclear transformation as well as chloroplast transformation. So for nuclear transformation, we can directly go for agrobacterium mediated, because they are very good at getting a recombination and get inserted into the plant genome. But when we go for chloroplastic transformation, we always have to go for biolistic methods. So one best advantage of using chloroplast transformation is we can get multiple copies of genes because of the presence of multiple copies of chloroplast. So higher the chloroplast, higher the transformation, and higher the protein expression and higher the yield. So that is one important method. So why scientists go for chloroplast transformation. So and then selection of the transformants, and then development of the plants, and then recombinant protein production, and finally functional characterization. So the transient expression, however, when stable expression is concerned, so we don't have to worry about purification. As long as proteins are produced in the plants, as long as the plants are growing well, so it's fine. Everything is fine. Whenever we want, we can just harvest them. We can then store them, and then finally we can go for purification, extraction and purification. But when we do transient expression basically involves very short period of time where we have to express our gene of interest, which is expressed in a very short period of time, and the product we are looking for is also produced very fast. So as soon as the product are produced, we have to isolate it, and then we should go for purification, extraction and purification. So how this is done and what are the different approaches they use for transient expression, we will see further. Next, the third part is suspension culture. So suspension culture, when they're looking for is they grow callus based methods. So callus are developed from single clones. So these single clones will have the same identity. So that is the reason all the genes will be expressed equally, and the protein production will have an impact. So that is why these callus are produced and converted into cell suspensions. So where they can be, since they grow in liquid media, they can produce very good expression systems. They can be a very good expression systems in producing our desired protein. So these are the different methods: stable, transient, and suspension based modes of expression systems can be done for production of these proteins. In stable transformation, they will just take both virus as well agrobacterium can be involved. These days virus vectors are more utilized because since they have high affinity for infecting plants, their promoters and enhancers are taken and ligated along with the transfer DNA. So this, when they do develop this kind of binary vector expression cassettes, they can do for multiple proteins, not only a single gene. At the same time, a plant can express multiple proteins with different kinds of desired products. That is one major usefulness of using these plant systems. And then second thing is plant suspension culture. As I told you earlier, so we can use tissues from different parts of the plants. We can induce the callus and finally develop suspension cells, so that can be directly used as a bioreactors. That can be cultured in the bioreactors, and then once the enough protein production is produced, downstream processing can be directly applied, extracted, purified, and then released into the market. So the maintenance and the maintenance of these suspension cultures is very, very easy. However, we should be careful to avoid any chances of contamination, because these are grown within incubated system. Chances of contamination will be very, very high. So we have to avoid that when compared to the field grown plants. And third one is viral vector mediated transient system. So this is one of the best method recently they have adopted, where they grow plants and then they also will develop these agrobacterium cultures. So once these plants attain a mature state, so they will be infiltrated, means they will be immersed into the agrobacterium suspension cultures, and then a vacuum will be applied so that all these agrobacterium carrying this desired gene will be imbibed into the whole part of a plant, and then the plants will be carefully grown in a particular incubation system, and after maturity they will harvest the biomass, and then extraction and purification will be followed. So basically green leafy vegetables can be targeted for such kind of production. So this is one of the best and easiest method. However, it should be these plants should be grown in a very controlled conditions. So yeah, the excess agrobacterium production can also lead to damage of the plant cells, so that should be controlled. So the next thing is advantages of advantages and limitations of molecular farming. So when we are discussing about different methods of transformation such as nuclear, chloroplast, and transient expressions, what are their advantages and what are their limitations? So nuclear stable transformation, high scale of capacity means large scale. Okay, we can produce them in a very large scale, and then glycosylation pattern will be good, and then low risk from the animal bone contaminants. So, and also they are very cost effective in nature, and several plant species can be used in producing these proteins. However, when we are producing any kind of vaccine, it is very important that we check the dosage. So we cannot just administer any kind of dosage we want. So for dosage standardization itself is very difficult. So that especially when we are going for edible vaccines. So dosage control is very much required, and it is highly cumbersome when we are using plants. And limitations of economic aspects of plant biomass production, including target of interest. Economic aspects, whether the target the product which we are going to produce is good or bad, what is the cost, what is the production amount, everything we have to take it into consideration. That is one major drawback when we are looking for target of our interested product. And when it comes to chloroplast transformation, high levels of expression, as I told, because of the multiple chloroplast organelles, when very high stability. Stability is very good in chloroplast, and open potential open cultivation by maternal inheritance. So since it is maternal inheritance, the targeted product will be produced in a high amount and very highly stable. The major importance of using chloroplast is homologous recombination will happen, and also the another criteria is silencing and mismatching. Everything can be avoided in this chloroplast transformation. That is why people go for targeting chloroplast. So, however, the major limitation is lack of research for commercial application. They are doing for the purpose of standardizing the protocol. However, for commercial purpose, still they have not yet been standardized. And then vaccine production can be limited only for some plant species, not all. So they have to do still lot of challenges are awaiting when they go for chloroplast transformation. And then transient expression, efficient and time saving, widely accepted strategy. So when we express in a transient way in the plants, you just need agrobacterium culture which is carrying a virus that has got gene of our interest, and just bring this syringe and then insert it into the leaves, and then allow them to grow. Within 3 hours to 6 days, the protein starts expressing into the plant system. It's very short period of time. So we don't need any kind of fancy incubation system, fancy equipment to introduce this gene of interest, anything. Just one syringe is enough. So, however, the effect of biomass and plant density, growth area, and leaf position plays an important role. So, the injection system and the position of the leaf where the point of entry is happening and the biomass of that particular plant growth area, everything will be contributed, and not every plant system can easily take up these culture inside them. So that is one of the major drawback. And yield changes in recombinant proteins are contributed by various factors. So various factors can be involved, such as like the temperature where the plant is going, the morphology of the plant, and then the virus itself cannot be expressed in the plant system. So these are the major factors. Some virus vectors may not be expressive, most expressive in particular plant system. So these are few of the factors that can contribute for the limited production of recombinant proteins. So next is, once we strategize the introduction of these desired genes for the expression of proteins, then comes the second thing is how to grow them, whether we should grow indoor or whether we should grow outdoor. So what are the pros and cons when we grow indoor or outdoor? These days you might be seeing even government is like endorsing, like go for vertical farming. There are a lot of usefulness of vertical farming. We can save time, we can save place, we can easily grow plants, and even they give good oxygen for us. It's kind of mutual benefit for both humans as well as plants. So one best thing is growing. When we are going for indoor cultivation, our choices like we go for greenhouse, vertical farming, bioreactors, and the best and foremost thing is hydroponics. Hydroponics is very easy, and these days soil can be easily altered, can be replaced by using hydroponics. So usually when we grow plants in the field, the availability of nutrients is very, very important. So plants have to strive hard for the uptake of these nutrients in the soil. But when we use hydroponics, it is in under controlled condition and all the nutrients are available, and plants can easily uptake all these nutrients and they can develop very good biomass. So when there is good biomass, obviously the protein accumulation will also be good and enormous. So that is the major use of using hydroponics. And bioreactors, these days you can see in biofuels and other major sectors, a lot of bioreactors, photobioreactors are used, where they can simply, you just have to set up in sunlight where they grow robustly, especially for algae and also some plants, tissues where they can grow and easily produce our desired product. So this is one of the importance of closed indoor cultivation. But however, open field cultivation, we can grow. We don't have to worry about like this controlling pH, sunlight, air, temperature, humidity. So nothing we need to worry. We just have to grow, go and plant them and allow them to grow and harvest it. So there are both pros and cons. So open field cultivation, obviously low infrastructure cost is required, and the rapid production capacity of expansion is very high. Higher the land, higher the growth. So the more realistic option for large scale production. So since there is no limited option for a place, the land, so we can easily grow them in very large scale and produce these our recombinant proteins in a large scale. But however, the major risk is potential risk of cross pollination. So government bodies, they will never agree to give approve these things if we are growing anything that is related to some pathogen related. So if it is kind of related to our other therapeutic, it's fine. But if we are using so in edible vaccines, what we do is, in normal conventional vaccine, we take virus and we heat kill or attenuate and then we introduce into our body. But however, here we use surface proteins of particular virus, which induces antibodies or immunity in that particular plant, so that we can easily intake and develop antibodies by ourselves. So that surface protein itself becomes a potential risk and causes cross pollination. They may go and contaminate other plants and it enters the ecosystem, and people who are in immunosuppressants, they eat it and cross reaction will happen and so many other things will happen. So this is a big major drawback when we are looking for open field cultivation. So that is why very strict guidelines have been given by the government, and they will not allow any kind of transgenic plant to grow. As in India, it's very bioethics, like they are very stringent and they will never approve. And only when we are looking for some other food related products, that is fine. But when we are looking for a therapeutic, it's highly difficult getting a nod from this bioethics committee. And then need to address GMO regulatory requirements and public concerns. Not everybody, if public will not approve this if they come to know. So that is why regulatory requirements are must and should. That is, these are the major drawbacks when we go for open field contamination. Because just see COVID. This COVID itself is just one small leak has caused devastation in the world. So that is why it's not just, we can do trial and error method, but putting ourselves in risks is not a good idea. So that is why we should be very careful when we are producing these pharmaceutical products. So next, coming to closed culture. What are the effectiveness or advantages of using closed culture? So they're very fast. They efficiently can be grown and optimized growth conditions can be achieved. And they're also cost effective and rapid scale, easy to maintain, and quality can also be improved, and high productivity can be achieved, and a lot of biomass can be developed, easily manipulated and handled. And then also we can prevent the hazards that are that happens in open field, such as through production of pollens or seed spread. Okay, so this can be prevented because they are produced in controlled conditions, and possible growth of transgenic plants expressing such transgenes. Okay, so these are the major advantages. However, the major impact is various factors can be contributed to contaminate suspension cultures. So we have to maintain them properly. The clones, they can get a lot of variations will happen. Physical, environmental, and biochemical variations can take place in suspension cultures. So constant monitoring is required, and that constant monitor requires a lot of investment. So possibility of contamination during subculturing, that is one major drawback. And bioreactor system, limitations of using large scale bioreactor system. When we are looking for bioreactor system, if we use higher large scale bioreactor, we can reduce the cost because we can use more at the same time we can harvest high large quantities of proteins. But when we are looking for small reactors, time consumption, media consumption, cell culture, electricity, everything will be taken into consideration. So that's why we have to be very careful. However, the larger the bioreactor, larger the problems we face. That is also because continuous will be there, batch reactors will be there, fed batch reactors will be there. So many things have to be taken into consideration. And then low commercial application of therapeutic products. Not many therapeutic products are produced using these suspension cultures. So that is why closed cultures have their own pros and cons when compared to open field. So everything should be strategically monitored and decided. And then this is about waste management. So one utilization or one best usefulness of using plants is we can avoid lot of waste. Suppose when we are growing plants in this bioreactors, especially these days, you might be aware of disposable bioreactors. They are made up of polypropylenes or different kinds of polypropylene. It's like a single-use equipment. You can just grow for that particular moment. After harvest, you just can dispose it off. But disposing it off, plastic waste generation, biological waste generation for a mere amount of product, and lot of cost investment, lot of time, energy, everything will be invested in when we are looking for cell-based culture system for production of these therapeutics. But however, when compared to that plant systems, if we see the proteins will be produced, some if there are any other extra things, they can be easily degraded and they can be converted into metabolites and then utilized by the plants in turn, and then biomass of plants themselves can be improved, and finally product can be developed. So in one system, one single user, suppose if we are producing 1000 liters of cell culture system, at a time nearly 61 kgs of plastics will come out as a wastage. But however, this doesn't happen in plants. 61 kilo plastics, if they become wastage, just imagine how much they keep on accumulating. So these are kind of different waste management systems when we go for plant, as well as it is one utility of going for molecular farming. So next is enzymes. So till now, what are the different kinds of industrial pharmaceutical products that have been released by the production of plants, which are produced from plants and has been released, or enzymes, avidine, beta-glucuronidase, inogen, lacase for production of bioethanol, paper bleaching, diagnostic kits, as well as pharmaceutical. These are the few of the products that are available, which are produced from plant system. And then vaccination. So when it comes to vaccines, a lot of edible vaccines are available, where bananas are major fruits that are used for production of these kinds of vaccines. For example, enterotoxigenic vaccines, which are produced these diseases called traveler's disease, that is caused by E. coli. So for the antibodies can be produced for such pathogens using potatoes and maize. Norovirus, where they express capsid protein in potatoes for that particular virus. Hepatitis B virus, surface protein has been expressed in leafy vegetable as well as storage plant. Rabies virus, and then influenza, cholera. So all these plants are commercially produced, and then can be are shown that edible vaccine can also one good option. So especially these things are given in developing third world countries like African countries, where they cannot afford a normal vaccine, conventional vaccines. So that is why edible vaccines can be a good source for people in such cases. And this is the first therapeutic pharmaceutical product that was produced in plants using carrot cell suspension cultures for a disease called type 1 Gaucher disease, and then released and can be easily taken up. So and then finally, in a nutshell, plants can be used for forming in a molecular way. Since we use recombinant DNA technology, that is why we call it as molecular forming when compared to conventional forming. So it has got a lot of advantages like great potential to produce complex mammalian proteins that are of commercial value and medical importance, low cost production, stability and storage can be properly maintained. That means you just have to freeze it and dry it and store the leaves, and whenever we need we can just isolate them, extract the protein, purify it and go for utilization. Next is safety. And there is we can avoid the contamination from animals and human viruses, pathogens, and easily reproducible in consumable form in the form of edible vaccines using leafy crops. That is, lettuce is a very good ideal crop for producing, and then these vaccines and fruits and vegetables. But however, the major problem here is biosafety issues. So gene and protein pollution. So these genes that are encoded to produce proteins can spread into the environment, can affect the non-target organisms. So if we are target organism, they can also cause contamination for other animals. So we totally disrupt the ecosystem, that we should avoid it. And then as I told, immunosuppressant people can also be affected. That one safety concern, we have to keep it in mind. And then finally, product safety. Concerns about such products should be harmful in the human or animal patients to which they are administered. Okay, so we have to see the background of the patients, whether they are allergic or not. So because some vegetables, some fruits can be allergic to different kinds of patients, we cannot just, without knowing their background, we cannot. Recently, one of our I was really surprised. One person, he was also a teaching faculty who had a COVID, and he took medicine for that. So minimum 90 days to 120 days he should have waited. Even before waiting that, he went and took vaccine, which get cross contaminated, like cross reaction happened between the medicine and this vaccine, and he lost both his kidneys. So all these things will happen. So that's why we need to keep all these things in mind before administering this to patients, because the glycosylation methods depends from plants to animals. So not everybody will be comfortable, and many are allergic. So everything should be kept in mind, and then we should go for harnessing the ability of these plants, which can produce these proteins in large quantity. So thank you all for listening. So this is my last slide, and I'm going to stop here my session.
Yes,
Yeah.
ma'am. So is there any questions from the audience side? They can unmute and ask if any.
Yeah. Excuse me, ma'am.
Yeah.
Yeah, I have a few questions, ma'am.
Yeah.
Yeah, ma'am, we are discussing about the vaccines which are extracted from plants as well as animals.
Yeah.
So we are extracting vaccines both from plants and animals. So according to you, which vaccines are better?
Obviously, vaccines from mammalian systems are better. So that is why even the vaccines, after isolating from plant system, scientists have to check whether the glycosylation, protein folding, everything is properly happened, and then they will be administered. But in mammalian system, we don't have to worry, because cell system, we can directly isolate them and extract them. So that is the one of the major limitation when we are really looking for plants.
Okay, ma'am. Ma'am, another doubt I have.
Yeah.
Ma'am, how do plants engage with beneficial microorganisms while at the same time restricting the pathogens?
Yeah, this is very important. Like pathogens, you see, these plants, it's quite complex. They will have their own defense mechanism. Okay, so you might be aware of silencing mechanism, right? So every day the plants keep encountering lot of pathogens in nature. So they will have a wide, there are so many defense mechanisms, still they're unraveling. So they have their own ability to control or suppress these pathogenic organisms, and also they keep producing. So since they are like factories where they keep on producing the different kinds of products in them, so they will develop different defense mechanisms which can overcome the pathogenic attack.
Okay, ma'am. Another thing, ma'am, how do pathogens evolve novel virulence activities?
Novel virulence activity. Pathogens, you know, the main problem, see, virus, for example, COVID went, now Omicron came, right? So different strains are coming. How are these all happening? So they are evolving because of this, like change of gene positions, change of gene virulence. So because of that, they keep on, they have to survive in one or the other way. So that is why they keep on, you know, these transposable elements and all those things. So they will make themselves more virulent by having positional effect of the genes.
Okay, ma'am.
Yeah.
Okay, thank you, ma'am.
Thank you.
Ma'am, another thing.
Yeah.
So in the tissue culture stuff that we spoke about, like
Yeah.
what is the efficiency of the plants sustaining their life when we take away them from the in vitro condition and after hardening to once it starts growing? So is the efficiency same from staying in the in vitro condition as well as to hardening, ma'am?
See, tissue cultures are just like test tube babies. They have to be like taken care of very well. I don't tell you, like, I don't give 100% efficiency will be there. And whatever 100 plants you take out from tissue cultured grown plants, 100 of them will survive. No, I won't give that. But 50% too, they will survive. So it all depends, because you have been growing plants in liquid, in a media where you are giving a ready-made nutrient solution, right? So they grow very well when you are giving everything, but when you put them in soil dynamics is totally different. Plants have to do circus to get all these plants. Lot of constraints will be there, where each, suppose one nutrient will be there. So plants have to recognize the root should go, chemotaxis should happen, the nutrient should come near. Plants have to release chemicals so that they dissolve those chemicals and absorb the ionic part. So many complex mechanisms will be happening in the soil. That is why acclimatization of these tissue culture grown plants will take a lot of time, and not 100% will be successful. So 50% plants can be successful, that I can guarantee.
Okay, ma'am.
Yeah.
So assuming that there are no further questions from the audience side, well, thank you very much for engaging the session today, ma'am. It was really nice interacting with you. And hopefully the molecular forming that you spoke to us today, it's really nice, and hope we can continue some research on the same plants again. So thank you a lot for engaging the session. And thank you, ma'am. Thank you again.
Thank you, everyone. And thank you, Tanishri, for giving me an opportunity. You know, I did this at the nth moment, and this is my first seminar I am giving. I was so nervous. But still, I hope you understood something. I'm not like 100% here, and I did not give my best, I feel. But I hope you understood something of this molecular form. And
Yes,
thank you,
ma'am.
one and all.
Yeah, thank you, ma'am. It was really good.
Thank you.
Okay, ma'am. Thank you from Nakshatra side as well.
Thank you. All the best wishes for Nakshatra.
Thank you, ma'am. Thank you so much.
Yeah.
Bye, ma'am.
Yeah, bye.